Hello, everyone. My name is Sarah, and I'm a postdoc in the Cancer Genomics Group at Southampton University in the UK. And today I'll be sharing with you how I've been using nanopore sequencing to study the FcγR receptors. So I'll begin with just a bit of background. Monoclonal antibodies now play a fundamental role in cancer therapy alongside the traditional chemotherapies. However, many patients still face persistent challenges of resistance and relapse. The success of these monoclonal antibodies depends on their ability to engage with a specific family of cell surface proteins called the FcγR receptors. There is one high affinity receptor and 5 low affinity. They all have activating signaling capacity to induce an immune response, except the inhibitory FcγR receptor 2b, which when bound initiates an inhibitory signaling cascade that dampens down the effects of any activatory signals. So this protein family plays a key role in immune defense, coordinating the elimination of pathogens and infected cells through mechanisms such as ADCC and ADCP. But they are also crucial for monoclonal antibody immunotherapy, where the balance of expression between the activating and the inhibitory receptors shapes a successful outcome. So we want to investigate what regulates their expression, both in normal and malignant settings. But any sort of omic study into the low affinity FcγR receptor locus is severely confounded by the inherent complexities of the region. So the low affinity FcγR receptor locus lies within a 200 kilobase region that contains a large segmental duplication with extremely high sequence homology, where these two shaded bars share nearly exactly the same sequence. And this creates a real challenge for your alignment software, as it becomes almost impossible to determine the origin of the sequencing read if it's just 200 base pairs in length. And this leads to regions that are consistently unoccupied, often called dark or camouflage regions. And then to further complicate matters, the locus is also littered with single nucleotide polymorphisms and harbors copy number variation, which can both alter protein expression. And these 4 CNV regions, or CNRs, show which series of genes have been reported so far to be duplicated or deleted, but due to the high homology, we just are still uncertain about the exact breakpoints. So we'd like to investigate the regulatory mechanisms behind FcγR receptor expression and the influence all these variants have. But because of the sequencing issues, we just don't have the genomic information to understand their functional impact just yet. So our solution is to use Oxford Nanopore sequencing, and I'm assuming this audience is already pretty well versed in the tech, so I won't be going into that too much, but I will say the flexibility of this platform is a huge benefit, and our key motivation for choosing this is its capacity for generating those ultra long reads, increasing the chances of spanning our tricky region. Plus, since it sequences the native DNA directly, you can also pick up on those base modifications too, as a little bonus. So I began with a largish cohort that had been subjected to FcγR receptor MLPA assay, a method commonly used to infer some of the well-known FcγR receptor variants, although very prone to ambiguity because of the complexity. Then from here, I selected 22 cases that were suspected to present a diverse range of FcγR receptor SNP and copy number states. And since the start of the project, I've tried out a range of nanopore preps and different devices with the aim to enrich for the region and to keep up with the tech's rapid evolution. Each of these produced a different level of coverage, and ultimately they've complemented each other to enable us to gain information on the phasing, sequence variation, and methylation. The large structural variants were confirmed by nano for the majority of these cases, and we've taken some of these forward for transcriptomic analysis too. So the first approach of optimizing a whole genome prep achieved an N50 of around 40 kb and confirmed these long reads were capable of uniquely mapping, successfully covering regions that are normally inaccessible. And then I spent a considerable time optimizing a few guide RNA designs for the Cas9 enrichment kit, which really dramatically improved the depth per run. But as I was designing primers to a very homologous region, this really complicates things, resulting in very uneven coverage and a huge pileup of much smaller reads. So I was very happy to move on to adaptive sampling, where I could enrich my libraries computationally. And this showed that we could capture those long reads from the whole genome prep while also ensuring nice even coverage across the entire target. So after all this data generation, I've now got a high depth of long reads, and this means I'm now able to phase across the entire locus in a single continuous block, which enables all the SNPs to be genotyped with haplotype resolution, providing insight into which variants tend to be inherited on the same allele, and potential linkage disequilibrium relationships. And this makes it possible to explore specific SNPs of interest in greater detail. For instance, these 5 polymorphic positions in FcγR receptor 3b define the clinically significant H&A haplotype, which alters IgG affinity and modulates effective phagocytosis. However, accurately determining the haplotype is very challenging with standard methods, as each individual SNP must be correctly resolved, an especially difficult task given the near identical sequences of 3b and 3a. Yet with Nanopore, it's easy to identify which combination of variants are on each haplotype, enabling confident inference of exact allotypes, which I found is often discordant with what the standard MLPA assay suggests is present. Likewise, we can also look at regulatory SNPs. In this region are 2 well-known polymorphic positions that define a promoter haplotype which impacts the expression of 2b and 2c. But accurate genotyping has long been a challenge. Nanopore sequencing now makes it straightforward to resolve these haplotypes across the highly homologous genes. And importantly, it opens the door to investigating a broader set of potentially regulatory variants whose functional effects may have been missed or overlooked due to previous technical limitations. And beyond individual variants of interest, Nanopore enables us to appreciate the entire FcγR receptor locus in its full context, something previously out of reach with standard short read or targeted primer-based approaches, which fragment and obscure the bigger picture. This not only reveals regions that might have been misassembled within HE38, but also highlights consistent differences across individuals, giving us a more accurate and complete picture of the true genomic landscape, including variants within intergenic, non-coding regulatory elements that have long been overlooked. Then on to structural variants, which is a bit more complicated. When using structural variant callers, the large copy number regions which we know are there could not be detected, and they only consistently and reliably identified these 8 polymorphic regions of short tandem repeats. And this is likely to be due to a combination of factors, but essentially our reads from the large A to KB duplications are aligning seamlessly back to the original segmental duplication, leaving no clear, consistent signal for the caller to detect. So we've resorted to assembling the region de novo. So we've been working to optimize the assembly process for this homologous region so that we can now move beyond relying on the structure of the current HE38 reference, which conceals the CNRs. But this is proving difficult for some cases with complex variants depending on which assembler or parameters are used, as a lot of them aren't well suited to deal with these large nested repeats. So while our goal is to achieve a lovely single contig per haplotype, we've encountered everything from completely fragmented assemblies to collapsed versions of the segmental duplication, and even just circular contigs. But we are getting there, building highly accurate haplotype resolved assemblies that provide base pair resolution for cases that are CNR neutral and those that contain a CNR duplication or deletion. And this is proving especially powerful for characterizing novel chimeric genes and promoter elements when the breakpoints fall within intergenic regions. As well as revealing more about the genomic landscape, we've also been able to explore the transcriptomic side, giving us valuable insight into the transcript diversity and important alternatively spliced transcripts. For example, there are lots of contentions around the expression of 2C, yet by looking at the full length transcripts, we've been able to see that many retain their final intron, introducing a premature stop codon 46 residues in. And finally, we're also exploring methylation in a haplotype resolved way too, as Nanopore makes it possible to get methylation data straight out of base calling with no extra prep time or costs. Once we have high quality assemblies, we're able to map reads with their molecule-specific methylation marks back, giving us an unprecedented view of regulatory modifications and how they may influence expression. So in conclusion, we finally have the tools to untangle this complex region, capturing a comprehensive haplotype view of SNPs, structural variants, and methylation in a single data set. And this unlocks new opportunities for ourselves and the broader community to carry out multi-omic studies on the region, supported by a single, unique resource for interrogating gene expression regulation. And more broadly, lays the foundation for ultimately advancing risk-adapted patient stratification and improving outcomes. And lastly, just a big thanks to everyone involved, and of course you for listening, and I'll be around at the conference or happy to connect online if you'd like to discuss any more. Thank you.